আচ্ছা, আচ্ছা। ঠিক আছে। এটা তো আমি একটু করবোই না। আচ্ছা, ঠিক আছে। আচ্ছা,
Hello everyone, I welcome you all for this national level webinar on research trend analysis of COVID-19 with the approach of scientometric and multidisciplinary applications. So please wait up to 3 p.m. So we will start the whole session from 3 p.m. Thank you. I thank you all for your patience.
Good afternoon, everyone. We are all waiting for people to join in. We have a lot of registrations, a lot of participants. We have our speakers today. This is just gearing up for the final session. As you already know, we will start sharp at 3 p.m. We a request from the entire Maharani Kasiswari College team. We request you all to please keep yourselves muted at all times, and your videos should be switched off for a better experience. I'm sure you already know that, but just a request from us. We will shortly begin sharp at 3 p.m. I extend a warm welcome to everyone who have already joined in, and for those who are waiting in the waiting room, Zoom waiting room. As soon as we have all of you with us, we will start off. We will start the program sharp at 3 p.m. So just 8 minutes to go. We are waiting for all participants. Our speakers, I think, are already in. I request all of you, those who have joined in a little later, to please keep yourselves muted and your videos switched off. We are just about to start in a few minutes. I request everyone to please keep yourselves muted at all times during the webinar. And please keep your videos off for a better experience of the entire webinar. If you want to ask a question or you want to maybe share something, you can use the chat box that is present below your screen. If you're using your phone, you will have it on the right side. We are about to start our program in a very, very short while. Uh, Thank you so much for being with us today. It's really nice to have so many of you on a Sunday afternoon. Our speakers are here with us. We are just waiting for a few more people to join in. We are all ready. Please keep yourselves muted, your videos off for a better experience of the entire webinar. You can use the chat box to communicate. Yes, I can see someone doing it right now. Great. So you can use your chat box, which is there down below your screen, or maybe on the right side if you're using a phone. It is really nice to see our speakers are with us. Our participants are also with us. We are just waiting for a few more participants who are probably having a little problem with the network. So our technical team is helping them. Really nice to see all of you here today on a Sunday afternoon. It is really nice to see our speakers are with us. Our participants are also with us. We are just waiting for a few more participants who are probably having a little problem with the network. So our technical team is helping them. Really nice to see all of you here today on a Sunday afternoon. We have our speakers with us today. We are extremely happy to have both of you today here. We are just waiting for a few more minutes. I request everyone to please switch off their videos and to please keep themselves muted. We have our speakers with us today. We are extremely happy to have both of you today here. We are just waiting for a few more minutes. I request everyone to please. Good afternoon, all. I think you can hear me. The sound is audible. I think so. So a very good afternoon. On behalf of the entire Maharani Kasiswari College, I extend a very warm welcome to all of you. I'm Dr. Ronindita Ray Chakravarti of the Department of Food and Nutrition of the same college. It's with immense pleasure that I extend a warm welcome to everyone to this webinar on recent trend analysis of COVID-19, a multidisciplinary and scientometric approach organized by the Department of Library of Maharani Kasiswari College. I especially extend a warm and a hearty welcome to our speakers, Dr. S.K. Sarif Hassan, sir, we welcome you all, and Dr. Vidyarthi Datta, who's also here with us. Sir, welcome to our webinar today. And it's with real honor and pleasure that we present to you this webinar and the Department of Library and the technical team, they've worked immensely for over the past few days to bring such an important topic to all of you today. And we have been joined today by scholars, faculty members, students, and people from various other fields from all over our country. Especially, I must mention that we have a lot of people from south of India, the peninsula. So welcome to all of you. And we also have a lot of people from the different other states as well. Special mention must be made of our participants from Indonesia and Tanzania as well. So it is going to be, we are hoping that it is a wonderful webinar today. And I think I should hand over the proceedings to Onindo. But before that, a very short brief up that I think that I should be telling you all who have joined in late, that we request you to keep yourselves muted, your videos switched off for a better reception. The webinar would be divided into two sessions. The first one on recent research developments on COVID-19, research challenges with respect to biology and non-biological fields. And the second session will be on scientometric analysis of citation data on COVID-19 research outputs. I think both the topics are extremely interesting, new, and all our participants are going to have a wonderful time. I think Principal Madam has also joined in, and with pleasure and honor, with a lot of pride, I welcome our principal, Dr. Shima Chakraborty, also to this lovely afternoon. We will start off very soon. I think she is connecting. As she is connecting, I would like to hand over the proceedings to Onindo Basu from the Department of Library, a convener of the webinar. So Onindo, over to you.
Madam, to formally open the session and give a short speech. What will you, madam?
I think there is a little bit of a technical problem, maybe because this entire thing is dependent on network. So if Onindo is ready to go, I request Onindo to take over. Yes, Onindo. I think there's a bit of a sound problem somewhere, Onindo. While Onindo is trying to connect, let us speak about this particular topic, the recent trends in research. There are so many webinars that are going on, but hardly in fact, very few of them are actually speaking on the recent trends on research and how to go about it and how the citations are helping people. So you see that this webinar is going to have a lot of impact on the present scenario. I'm sure you all will agree with me that, and the speakers obviously will speak on this. I am not a person who is equipped to say a lot on this topic. But I'm sure that once Dr. Hassan and Dr. Vidyarthi Datta gets over with their sessions, I'm sure there will be a lot of questions, there will be a lot of insights, there would be a lot of discussion on the topic. And this, I think, will help us in a very long way, help us to go on, in a very long way and help us all in our research. So I think Onindo is all ready. Yes, Onindo? Over to you again.
Good afternoon, everyone. Department of Library of Maharani Kasiswari College has organized this webinar on research trend of COVID-19. Being the convener of the webinar, I just want to give a short briefing stating the necessity of this theme. While spreading of this virus has jolted every scenario of social life. Sudden outburst of the pandemic has also ignited the research front across disciplines. Similar event happened after World War I and more specifically after World War II. Exponential growth of scientific researches happened right after World War II. But COVID-19 itself has become such a vibrant yet a very nascent domain which is attracting researchers from different subjects such as medicine, biomedical science, genetics, economics, virology, pathology, sociology, education, statistics, mathematics, international relations, and whatnot. Within few months, global scientific community unites to track the progress of COVID-19 research and development, identifies new research gaps and priorities. Developers and funders from all over the world, all of whom shared approaches and raw data freely. In a show of solidarity from the global science community, all major research institutes carrying out trials shared their data with a view to speeding up scientific discovery and implementation of the solutions. It is worth mentioning that the common people, researchers, as well as the teachers are now understanding the necessity of open access to technologies, learning materials, and research deliverables. And we are embracing the ethos of open access more than ever before. Every health guideline in recent times being issued by any authority regarding COVID-19 to the public is always backed by the research evolved on trial and run basis. Scientists are toiling hard day and night to understand the nature of crisis in different dimensions and struggling hard to find optimal solutions. Within the process of trial and run, different hypotheses are coming up. Yet again, some of them are getting sidelined due to initial research breakthrough and cross-validation at later stage. For example, the usage of hydroxychloroquine and later being banned by different health agencies and policymakers. Another example is whether COVID-19 is airborne or not. So we have to understand that a research, even if it is validated by data and techniques, may get nullified or rejected by another experiment in different settings and environment, or change of population or sample, or change of granularity or change of complexity, as well as the change of parameters. This critical deviation is very costly for human lives. So many research gaps are visible, so many questions are being generated, but most of them cannot be answered, which openly declares the demand of more and more researches with multidisciplinary collaboration. All of the people are looking at the research of vaccine, but we don't know whether this vaccine will be effective for all people residing in every possible weather conditions or not. We don't know. How long countries can sustain economic recession? We don't know. While what will be the mode of education in future? We don't know. Whether a vaccine will be effective over every possible genetic mutations of COVID-19? We don't know. What will be the geopolitical situation, capital crisis, or post-traumatic stress symptom of COVID-19 patients in near future? We don't know. The more is the questions, the more is the opportunities of researches across domains. In Indian context, less than 3% of GDP is spent after education, and this is the perfect time to invest more. International, sorry, information professionals have unique opportunity to critically think and develop techniques on research duplication, data sharing platforms, design of metadata for data, critical indexing and abstracting service to research articles, etc. So it is our perception that at least for next 10 years, researchers will continue to do research in this area. The resource persons who are experts in their respective fields will enlighten us with the trend of researches, open problems, progression of research, and other related issues pertaining to COVID-19 only. Unless we know the growth and progression of research, information services cannot be provided precisely. Hope you all will be benefited and enriched by their inputs. So I am now requesting Orpita Bhaduri, Assistant Professor of Computer Science, to introduce the first speaker of the session. Thank you all. We want your active participation till the end and contribute some by asking questions at the interactive session. Thank you.
Thank you, Anindada. I am giving a brief introduction of Dr. S.K. Sarif Hassan. Dr. S.K. Sarif Hassan is presently working as an assistant professor in mathematics at Pingla Thana Mahavidyalaya. Prior to it, Dr. Hassan did his postdoctoral work from the International Center for Theoretical Science, TIFR Bangalore. Dr. Hassan has completed his PhD work from Applied Statistics Unit, Indian Statistical Institute, Kolkata. He has worked in fractal geometry, discrete spaces, integral value transformation, mathematical morphology, data assimilation, mathematical genomics, computational algebra. He has authored more than 80 research articles published in prestigious journals and conferences. It is worth mentioning that one of his works on Y factory genome got published in very prestigious Nature journal. His latest research paper on changes of SARS-CoV-2 virulence property in Indian patients has opened a new research area for the scientists working on COVID-19. Thank you. Over to Dr. S.K. Sarif Hassan. Thank you.
Thank you all, and it's really a great pleasure to be here and invited by you. And thanks for the nice introduction, and to the organizers, the team, for putting these things in a positive effort towards discussing the things. Although I am not a expertised in this area, I mean, I'm not expert in this area. I mean, I'm not a biologist, I'm not a virologist, I'm a mathematician as mentioned. So this is a high time we must investigate these things from all of our respective perspectives to understand what is going on and why is this pandemic is we are all experiencing very badly, and it's something we don't know really where actually we're heading to. So in this very difficult time, it's really required to discuss among all of us and what could be better scenario. As Anindo said, there are several issues. I should say it's every field has different new issues centering this COVID-19, health issues, education issues, even life-threatening things we are experiencing day by day, and it's very much scaring things. So on that light, what I would do is I won't go to very deep things or discussion, because I understand there are various people having various expertise or various interest of different disciplines. So I talk mainly in brief what are the things going on, and to the best of my knowledge, of course, and possibly that would give you a kind of flavor that what's going on, and then you would get fascinated to work on this kind of aspects, as I understand, and that's how I get into it. And hope that we can forward it to. And at the end, we can of course discuss the things. The things I'm going to talk about. Wherever you feel difficulty, please note it down, and we can talk those things later. So I'm just sharing my slides, and then we'll talk. I hope this is visible to all of you. Can anyone please respond, please, that it is okay? This screen is shared. If the screen is well shared?
Yes, it is shared now.
Okay, thank you. So let me talk. So mainly, as Anindo was telling, that we must understand what are the research currently going on to understand better things to make it forward and so. So within this very short amount of time, I mean, I would say from January onwards, we have extensive research on these centering this COVID-19. So some of the things I am going to talk about, and broadly I am putting the things in place to understand that how we should think in what are the directions we must move on. So those things will actually concentrating on it. Sorry. So what I have planned to talk is to introduce COVID-19, and there is no room actually to introduce this COVID-19. It has already introduced and ruining our society and entire nation. But still, the technical introduction possibly I would give, and there is no harm if we repeat, if you know. So that I'll do, and then I'll talk within a very short history that what are the key events of COVID-19 we are experiencing. This is a very dynamic phenomena we are having. So I'll just put into some points that these are the key events that we have encountered so far. And then I'll talk about the virus, which is actually causing this particular disease, COVID-19. So that the structure I'll talk. And there are so many controversies on origin of this particular virus. Still it is. And so there is something called genetic origin. That means it is there is a kind of evolutionary connection of this particular virus. So that aspect I'll talk a little bit. And then the immediate primary questions which comes in mind. So those things I'll discuss. And these questions are not not from all aspects are there. I mean, this is a very active primary problem which we must address. Those are the questions I'll focus, and there are many more actually. So those things I'll talk, and then I'll talk about the nature of this particular virus. The virus is called SARS-CoV-2. I'll tell you, this is called severe acute respiratory syndrome coronavirus 2. Because 2, why I'm saying 2, because earlier we also had this coronavirus. It is very long past it has actually. So it's 1938 when 1968 when the first human coronavirus was detected. So this is possibly the evolutionary generation of that virus, of those virus. So I'll also talk about the present research things which are going on. And on origin, there are research also. So those things also I'll talk. And I'll also explain genomic aspects of the virus, because it is all about that. So those things I'll also talk about. And then I tell that why non-biologists also should be concerned about COVID-19. Of course, medical practitioner, drug designer, core biologist, rather molecular biologist, they are all thinking very actively. But why then we also being non-biologists to think on this? So this is actually absolutely important, those things I'll tell you. And in a very personal perspective, I'll tell that what are the key subject or drivers, I would say, to drive over this virus. Those things I will also talk. And I'll very briefly, I'll talk about and work on a particular protein called ORF3A. This is called an accessory protein, which is involved in the virus SARS-CoV-2. So I'll very small piece of work I'll talk, which is recently worked with our team and it is published in Genomics. So, and then I'll talk about the present scenario of research, what's going on and what volume actually we are having nowadays, and what are the database and sources available for this COVID research, and the keywords, the new keywords, and almost all of us have so many new keywords: social distancing, asymptomatic, all pandemic. These are the kind of new keywords are known to all of us almost. So those things I'll put into it. And then issues on extensive research. And there is something called issues. When we sometimes say that we need extensive research. Indeed, we need. But there are issues whenever you have that extensive amount of research. So those things I'll tell. Even within this amount of time, we have those issues actually, and very severely. So and the kind of suggestions and initiatives that are taken, those things also I'll tell you. And at the end, being a layman, I'll put some non-technical suggestion to improve these issues, rather, I would say. And at the end, I'll put in a positive word. I'll try to say about vaccine, as Anindya was telling, that there are a lot of issues involved in this particular thing, and we're all eagerly waiting for it. So I'll put in a positive form regarding vaccine. So the COVID-19 is, as I said, it's a coronavirus disease. It's an infectious disease caused by newly discovered coronavirus. There were when coronavirus, as I said, previously coronavirus 1, we called it as SARS-CoV-1, and now we call SARS-CoV-1. Earlier it was just said SARS-CoV because there was only one. And previous to that, there was MERS, Middle East Respiratory Syndromes, and there are previously other bat coronavirus, pangolin coronavirus. So this coronavirus can affect also in other humans, other animals also. So those things are there. So the epidemiological data suggest that COVID-19 has spread widely from the city of Wuhan in China. So that's the kind of assumption you can talk we made, and this is kind of well accepted that this is the origin, epidemiological origin. And we all know what are the symptoms. Most people infected with COVID-19 virus will experience mild to moderate respiratory illness and recover without requirement of special treatment. I mean, nowadays, what you have is not having any medicine towards this coronavirus. We are not giving it. Hospitals or medical facilities are not towards this. It's something like you can boost up your immunity. So that's what we are trying. We are trying to make us healthy in so and so form, healthy, whatever you mean, as you understand. So that is what is the prerequisite to even being positive to negative. So that is the thing we are actually supporting. That's the kind of support system we have, and that's how we are recovering also. So that's the good sense of it. So that's how we are going on. I mean, itself, it doesn't require much special treatment. But to prevent, we need vaccine. And the virus, which is actually causing this COVID-19, is called severe acute respiratory syndrome coronavirus 2, and that we call next onwards SARS-CoV-2. And these are the key events we have. On 31st December, Wuhan Municipality Health Commission in Wuhan City clustered 27 pneumonia cases of unknown etiology. That means you don't know the origin of this. I mean, you don't know the symptom of this. You don't know the infection mechanism where from it is. So those things are unknown to you. And that's how 27 cases were identified in 31st December, at the last date of 29/20. So then on 7th January, identification of the novel coronavirus. So this is the new virus that is identified on 7th January. And on 9th January, China CDC, Drug Control Board, reported that virus had been detected at the causative agent for 1559 pneumonia cases. And 10th January, the first genome sequence was made and publicly available in the NCBI, National Center for Biotechnological Information. You see how rapidly the sequence is being made. On the first day, the first primary task is to put to the public that what is the sequence of the virus. So as I was telling, the genome sequence is very much important because that is the primary basic structure of the virus. So that was done and given. And on 10th January, as I said, and 13th January, the first case of infection outside China was detected. And one was from migration from China to Thailand. And Chinese authorities confirmed that the human to human transmission. That was the danger part was. And on 30th January, WHO declares the public health concern. It is an international public health concern. So from 13th January officially, it is actually affecting people or so, whatever you call. So these are the key events. We must know the past, and this is the key past of it. And the image you could see here is the SARS-CoV-2, and it is this is the kind of microscopic view of the virus. And if you cut it, I mean, if you take a kind of projection, two-dimensional, then you could see the right-hand side image where the proteins and all these things are marked. There is something called spike protein, M protein, membrane protein, and envelope protein, E protein. So as I was telling, envelope protein. So these are something called structural proteins. So they are embedded in the virus. The outlier things are there. And there are many other proteins associated to this virus called ORF3A. ORF means open reading frame, and 3A is a kind of name you have, and ORF6, ORF8, ORF7A, 7B, ORF8, ORF10. So it's an assemble of many proteins, around 11 protein you have. And earlier also similar number of proteins were there. And there is something called ORF1 also that I didn't mention here. That's a kind of polyprotein. You have there around 16 proteins. So altogether you have around 29 approximately. In SARS-CoV-2 it is about 26 proteins you have. So these are the proteins I mentioned here: spike, envelope, membrane, nucleoplastid, ORF3A. These are very key proteins. Of course, there are other protein in the ORF1 sequence. So there you have polyproteins, as I said. There is something called RDRP. So that's also a very important protein in virulence of this virus. So I'm not going to detail of those things. As I said, there are 26 such kind of proteins, and these are the kind of key proteins you have. And regarding genetic origin, you see, as I said, this particular virus is not new. This virus is new in some form. So this has a significant sequential similarity with percentage 96.3, 89%, and 82% with bat CoV, and the coronavirus which affects bat, and SARS-like-CoV. There are many others, and SARS-CoV, MERS, as I said, pangolin. So it has a sequential similarity with the previously identified coronaviruses, and which confirms the genetic origin. So that's the kind of accepted fact. It's a kind of published fact that is how we can expect that if it is kind of naturally evolved virus, so that is what is confirmed from their sequential similarity with the previous texture I am saying about the sequence. So the genomic sequence have this kind of similarity. I must tell you, if you are a statistician, don't ignore this percentage 96.3. You shouldn't think that, okay, so 96.3 is good number, good enough, close to 100. So we can think of this is very much close to bat wave. It's not really that. Even a single nucleotide make a havoc change. So those things possibly I'll talk, and how important those things that you will understand, because mutation is all about that. So what makes the new coronavirus so much more infectious as we are experiencing? Is it naturally originated or lab constructed? Although this question is very much not settled, and this is something is still controversial, and there are two different school of thoughts and having concrete logic, but only lab experiment, future wet lab experiment, things will disclose what is the fact, and we are heading towards that discovery. How significantly different from other previously identified virus? As I said, it has, but we must understand that how significantly different is this. How does the virus evolving? That is what is technically called mutating. I mean, changing its own character. by changing the nucleotide bases A, T, C, G to the other one. And I just must mention that this particular virus is RNA virus, positive RNA virus, positive sense RNA virus. This is once it get into the cell, host cell, it immediately change. Change to the protein sequence, the amino acid sequence, and immediately activating for further work and activities. So this is and this is one of the largest known coronavirus, RNA virus. The genome sequence is almost 32,000 long ATCG sequence you have. The entire genome sequence is of that line. So this is the largest RNA virus so far we know. And it has very special character, those things I'll talk. And the missense mutation meaning is that whenever a particular nucleotide is getting changed, if the associated amino acid is also getting changed and their kind of property, then you have functional changes. So if that happens, then you call those mutations called missense mutation. And there are many other kind of neutral mutations, there are nonsense mutations, there are several others. Molecular biologists, they know better than me. So that I'll talk that whom does it help, this mutation of the virus? Does it help to host or virus? So every virus mutates. This is the kind of nature you have, and it's a part of the virus life cycle. And those changes, shifts, are not always a big deal. As I said, there could be mutations which are neutral. It doesn't affect to either host or virus, but sometimes it is. So once SARS-CoV-2 makes contact with host, it starts to make new copies of itself that can go to infect other cells. So that's how it gets spreaded over the entire respiratory system. The new coronavirus SARS-CoV-2 has already mutated a handful amount of times within this very short amount of time, which makes people wondering whether the mutation could lead to a more severe or deadlier disease. We expect it is not, right? So we don't know, but we are investigating towards that. So accounting mutations over the proteins of SARS-CoV-2 and understand their consequences is certainly one issue we must all take up. And there is another thing, as I was telling, the spatial organization, the spatial arrangement of the proteins or gene and the whole genome of SARS-CoV-2. So that we also must understand, the spatial organization, how the nucleotides, how the amino acids are arranged one after another. So that organization is very much important, because you understand that the entire living, all living shells and living bodies are assembled of this basic thing called nucleotides, A, T, C, G. And this is the 4 content. And this is something very much interesting to talk, that to write English, to talk English, we need 26 letters to talk, A, B, C, D, like that. But nature has written everything within 4 things, A, T, C, and G, everything. So this is the basic content, and that's the total set of contents. So only thing, one gene is different from other, one virus is different from other, one mice is different from a human being, Onindo is different from me. So all these things are written in this ATCG sequence. So definitely the arrangement of ATCG is very much important, because content is same, even sometimes length is also same. But what is then different? What is the difference? The difference is the organization of the ATCG basic line. And after that, of course, it has other consequences in functionalities. But this is the basic ingredient, and their arrangement is very much important. And there is something called phylogenetic analysis. That is something called sequence-based analysis. I mean, how sequences are similar. Based on that, you can put an evolutionary connection. So that also, of course, one of the concerns that we must take care of. And finding phylogenetic relationship from other perspective, such as conservation of nucleotides, how the nucleotide bases are conserved over the sequence, how dimers means double, A, T, A, C, A, G, like this. So you have 4 letters, A, T, C, G, and you have suppose 2 option to take altogether. So A, T, A, C, A, G, like that you can take 16 such kind of possibilities you have. So those are called dimers, and codons are basically triplet of ATCG. So you have 64, 4 to the power 3, and amino acids, you know, all 20 amino acids are there. And there are some bases called purine bases and pyrimidine bases. So their special organization and conservation is also important to take into account to have the phylogenetic relationship of the viruses. So the other aspect is understanding the infection mechanism, which was very much difficult, because you see the virus having a different set of proteins, and when it get into a cell, host cell, it has another set of respiratory proteins, as I am telling, because this is involved in respiratory organs. So you have this, and so 2 different cloud of proteins are interacting, and a kind of new functions is happening. That is what is infection. So this mechanism is very much difficult, very much complicated actually. So the interaction between host protein and the virus protein is tremendously difficult. I don't know how nonlinearly we can think of, but it is much nonlinear than that. Nature is like this. So that we have to understand. And this is not difficult just for me being a non-biologist, even difficult to a virologist also. Excuse me. So then I'll, of course, this is another concern, that the virulence and infection control. How would you control it, and how virulent the virus is? Those things are also of another aspect to concern. And drug design, as mentioned by Onindo, is important and ultimate desire we have. And the other, we must also know the source of this virus. That's also important, as I said. And of course, there is another aspect called modeling of pandemic behavior of COVID-19. We must understand that what is the projection or prediction of the number of infections, number of infected people, maybe possibly in 2021. So what is the projection, that also we must understand from this previous data or present data. So that modeling problem is another big challenge towards this issues. And transmission traffic, that where from this transmission is taking places within India, from a country to another country. So there are so many such kind of geographical, epidemiological transmission. Traffic also is another concern to understand, because that also would help you to classify the nature of the virus. And weather and all those things also come into play, because different geographical locations having different kind of climate. So whether those things are also in effective form on this particular virus, that's also important. So transmission traffic is also important, and many more things are opened, of course. And these are something active things I am talking about. I'll also talk about the passive research, passive concern, passive issues, as already mentioned. So based on genome analysis, SARS-CoV-2 is a virus that evolved naturally and is not a synthetic lab strain. This is already a discovered fact, and the article published in Nature Medicine in March, possibly March or April was published, but the news came in March. And the proximal origin of SARS-CoV-2, and they are saying that it is not lab synthesized virus. This is naturally made, and that's why we are nowadays concerned about the genetic origin, and we are heading towards that. But still, it's controversial. It turns out that most of the SARS-CoV-2 strain are more than 99.5% identical on a nucleotide level, as I said. So again, in warning to statistician, because it's 99.5. So don't ignore 0.5%. You try to understand how big is this. So 32,000, as I said, 32,000 long. And if you take this 0.5%, it is about, I think, 162 or 160 something, that number, 150 or whatever, this number of bases are different. And so that would lead to another 60 or something like that amount of amino acids. So that amount of amino acid is changed in a virus. That's huge. Molecular biologists must appreciate this particular fact. They know much better than me that this is a very severe changes actually. So this 0.5% is very much significantly important, that you all must agree, and with a special gravity we must see on it. So interestingly, at the whole genome level, the SARS-CoV-2 is nearly 91% identical to the pangolin CoV, indicating that pangolins could be an intermediate host, although it is not yet confirmed, because for confirmation we need wet lab, different lab experiments. So that is not yet confirmed, but theoretically, even phylogenetically, it has been seen. So what are pangolins? They are eating mammals. So they are in high demand in Asia for use of traditional Chinese medicine, as well as for their meat as a food, which may consider as a delicacy. So it is a kind of source of human for human being. This is a kind of source for food or source for medicine. So this is very much critical host, if this is true, that intermediate host. We also don't know the whether it is intermediate or immediate or some kind of parental host. We really don't know those things, but it is of course in the line. And this is the kind of snapshot of the sequential similarities of this particular virus. And SARS-CoV-2, as mentioned, is different from other known coronavirus with 88% or less sequential identity. And beta coronavirus, it's a big family having different coronaviruses, and typically they are called MERS, SARS-CoV-2 and SARS, which happened in 2003 in China. And there is another virus called alpha coronavirus. So this is a broad family you can think of. And if you do a kind of sequential similarity, how close they are by sequential similarity, so you see this is a kind of classification you could see. The SARS-CoV-2 is significantly different in the beta corona family. MERS and they are basically kind of exclusively disjoint, and SARS also. So you understand from the even sequential similarity, although it has a significant amount of similarity, but it is significantly different even. So we must understand the genome more closely that we are used to. As I was telling, we must not ignore 0.5%. 99.5% similarities doesn't give you a driving force that you ignore it. No, don't do that. So what is the difference of this genome that we must understand? How does it significantly different? That we also have to take into account to further research. It needs our special attention, as I said, and we must investigate or understand the spatial organization of these genes and genome. This is very much important, which usually, to my personal perspective, biologists, molecular biologists ignore. They start thinking from protein sequence, they start thinking from function of the proteins. But to my perspective, we must also understand the basic primary structure from where whatever you would see is function and all. But we must understand the basic structure. That is very much important. So for all living being, as I was telling, the content of a gene is A, T, C, and G. But if it is an RNA, then it's called AUCG. The thymine T is replaced by uracil, and 64 codons, 16 dimers. So these are the basic component of a gene or genome or protein. And 20 amino acids are the constituent of a protein. So as I said, to my limited knowledge, these are the kind of subject can be used as a driver to drive over this particular virus called fractals. Fractals means it basically give you a inner picture of the virus, inner details of the virus. And the outskirt you could see, right? But to understand deep the inner behavior, the actual characteristics, the spatial characteristics, you have to have this tool called fractal to investigate. And mathematical morphology is another one which is very much important to understand the structure of the sequential organizational structure, or maybe the surfacial structure of the virus. This is very much important. Sorry. And I know that Anindo is kind of aware about this subject, mathematical morphology, and he also worked on this with a great scientist on mathematical morphology, B.S. Dayasagar from ISI Bangalore. Anyway, so that's very much important as I understand. And spatial statistics, that's also important to understand the spatial features of this particular genes and genome. And of course, molecular biology is the handle. And information theory comes into play to understand the information theoretic perspective of the sequences. The conservations and all those things also very much important. So information theory could be a good handle. I mean, these are the kind of subject you can think of would help from different subjective perspective, like computer science, theoretical computer science, biology, molecular biology, phylogenetics from genetics point of view, and finite space dynamics. This would help you to understand the dynamical behavior of the sequence. I mean, when it does get mutated, so we must understand that how this genomic transition are being occurred. So that finite space dynamics also important. And there are many more tools one can think of to proceed further. So this is the work we did, molecular conservation and differential mutation on a particular accessory protein called WRF3A gene or protein in Indian genomes. So those are the first primary work we did on that. And this article published in Genomics. So WRF3A is the largest accessory protein with 828 nucleotides. This is very much deterministic in form. The role of accessory proteins are of immense importance in replication of the virus, regulation of infection of the coronavirus in the host. So you must understand that the accessory protein has an immense role in virus virulence. WRF3A modulates the host response to the virus infection, and consequently, it plays an important role in pathogenesis, which is very much important to understand. So this is one of such accessory proteins, and this is the largest accessory protein among all the accessory proteins, having 828 nucleotides. So in this study, what we did to decipher the conservation of nucleotides, dimers, codons, amino acids, as I was telling, is important. And we did this thing on a specific gene. Instead of entire genome, we took this particular accessory protein and we did study over that. And across 32 genome of Indian patient. And that is the number of data available on 14th May 2020. So we took all those data from NCBI database, National Center for Biotechnological Information. So from there we did this kind of analysis. That means 32 WRF3A gene you could get from all these different genomes, 32 genomes. So those things we have scanned and we have reported. And WRF3A gene possesses single and double point mutation. That means one particular nucleotide is getting changed in some places, some position, and there are two nucleotides altogether simultaneously mutating, changing to another two. So that is what is called double mutation, double point mutation in Indian genome that we have seen. And that suggests that the change of this particular virus virulence property of Indian patient. So within India, this virus is evolving and changing. That is clear, that is transparent from this very small amount of data. So we find the parental origin, mind it, parental origin of the particular gene over the genomes of SARS-CoV-2 and pangolin is same. We are not saying pangolin is the immediate host or intermediate host. We are saying the parent of SARS-CoV-2 and pangolin is same. That is what is clear from the phylogenetic analysis based on conservation and nucleotides basis. This is something is very much new. Usually, biologists are used to do the phylogenetic analysis based on sequential similarity. So as I was telling, sequential similarity, the percentage would not give you the entire picture. So when it is not that, then you must get into much more deeper insights. So these are something we understand the conservation of nucleotides from the very information theoretic point of view using Shannon entropy. Those things can be taken into account, and that is done. And thereby we basically conclude that the parental origin of these two things are same. That is what is reported in this article. And this is the kind of picture you have of mutation. So ORF3A type 1 I am telling, as I said, the 828 base long the sequence you have, and there you change whenever you have a change G2T, you see there is a, that is written in the arrow, the G2T it is changed at the 171th position, and that change lead to a amino acid change Q to H. So Q, whenever you have that, you call type 1, and whenever it is H changed, then you call it as type 2. And from type 2, another two such kind of changes also happened in Indian genome, mainly from Gujarat, and that is type 3 and type 4, two different kinds. It is mentioned, and these slides would be shared to you so you can have a look in detail. So what I'm telling is that from type 1, you have you have another type 2, and these transition happening almost everywhere in the world, Europe, America, everywhere, you have this type 1 to type 2 transition. But type 2 to type 3 and 4, this particular mutation is was very much specific to India. Still, even last night I also checked, this kind of mutation is not occurred anywhere in outside India. So this is a special mutation that we have encountered. And I must tell you, because this is a kind of investigation we did on 14th May afterwards, and this paper published in June, then also we checked, and even 2-3 days back also I checked for new mutation. We have new mutations actually over this particular accessory protein of new kinds, but they are not evolving from type 2. Some of them are evolving from type 2 to type 3, but some are evolving from type 1 itself. So different such kind of mutations are happening. And as we expect that even we are having so many such kind of mutations over a specific accessory protein, which have different specific roles in virus virulence and pathogenesis, we are not getting too much death rate. So that is the positive flavor we could get here. So that's why you are telling that this mortality rate has been, decreased. So somehow this virus is mutating itself and killing itself, rather, I would say, because this mutation is not affecting too much to a host. The virus is not much virulent after this kind of mutation. Roughly we could say that this virus is getting weaker day by day, having this kind of mutations, and this is the natural process. And this is the snapshot of the data, and I was telling that you see the marked the areas, the type 2 mutation we have, March onward, you see April, 26th April, 26th, this is the collection date of the sample. So April onwards, we actually detected this new mutation type 2. And you look at, even in April, we also have the type 3, type 4 mutation within this small 32 data. So now we have more, and we have more mutations, more different types of mutations we have. So you try to see within a very short amount of time, even within a single month, April, we have typical difficult mutations, and these are very much unique mutations that we have. We don't know why these mutations happened in India, why not outside India. So we expect that we are very much healthy, we eat various things, and possibly our immunity is better. So those are the things possibly is leading the changes, mutations in the virus, and that is not going in favor of the virus, that is going in favor of us. So that's how we are actually winning, you can say. So then, as I said, I did a sequential similarity based analysis as well as phylogenetic analysis, and we could see that this particular gene itself has closeness to the pangolin COVs. So I think this image is not clear, but you can see in a greater window better the sequential similarity is being depicted here. That is what is written here. I am not repeating these things because of time constraint. And So as I was telling, within this amount of time you have, as per WHO information, I mean, this is the update I did at 11 a.m. in the morning today. We have 43,322 articles, published article on COVID research. You can see the COVID-19 global literature on coronavirus disease. You have 43,322 and full text available 40,000. All these are marked. And there are almost same amount of, even more than that, unpublished preprints available. You see the huge amount of research. Now question, how independent these researchers are? So I was thinking that, okay, so if you write a paper, or if you write a review paper on coronavirus patient research, how many reference I should make at the end of this article? I don't know really. Possibly I have to mention 20,000 articles, even more, to do a good review, extensive review on that. And I know, and you can Google out, I was doing that what is the most cited, most having reference paper? On coronavirus, Wikipedia page is the most cited one, I mean, referenced one. It has around, I think, 1500 amount of references on COVID-19, Wikipedia page. So none of the scientific article can cover all these amount of references. So you see the extensive amount of research. We don't know really when you are submitting a new article to a journal, even journal editors and chief cannot conclude that this thing already has been done and submitted to some other journal. It's very much difficult. So it opens a new arena of data science, big data analytics, research on information science, information processing. So many opens thing we are having this extensive research. We don't know the use of this extensive research. Why? What happens? What next? What is the bottom line? What is the synopsis of this? I don't know whether any expert could make such kind of comment or not. So that's the danger thing we have. And Elsevier have a dedicated forum on novel coronavirus research directory. It has more than 30,000 articles, 31,000 research articles already published in Elsevier publishers. You see the amount of things, and you know there are so many prestigious journals are archived in Elsevier, including Lancet and all, having 50 impact factor. You understand the gravity of this particular journal, and having so many articles, in fact. So we don't know, we are in a cloud actually. And also you have COVID-19 Elsevier, another forum called Research Expertise and Collaboration. So that is another database you have, that who are working on this and what are the areas of those researchers. That is also made. These links are given at the end of this talk. So I'll share with you all. This is powered by Scopus. They're all doing, and you see this is the research profile, the 6942 number of researchers are working. I mean, within this amount of Elsevier's authors directory, and there are 1762 research units, and there are relevant research of about 23,000 and 191 data sets, and all this kind of data you have. So if you go into this particular database, you can search who are people working on particular on that and that areas, so that you can fetch and you can start collaborating, you can interacting with those people. So this is a very good forum is being created by Elsevier to do that. So the aim is already been declared here. Identify potential research experts on collaborators, areas related to the coronavirus epidemic across basic science, translational research, or clinical practice, which is what are important at this time. So these are the very few of important resources that I shared. This is the NCBI one. The first one is for the sequence database. You can go and download all the sequence uploaded even today. Day by day it is increasing. You can always keep on update. This is getting updated, so you can keep on tracking the data from India also, from various countries, everywhere you get this. And next strain, that another database similar kind, having sequences and protein sequences, also some the protein structure also, So those informations are given. Even in the GIS AID also have the same kind of information. So these databases are of that kind, as I was telling, and this includes all almost. So there are other secondary level databases also, but these are kind of primary database to the best of my knowledge. So these are the kind of keywords, even more actually. So we get that COVID-19, SARS-CoV-2, pandemic, mutation, asymptomatic, coronavirus, many more such kind of keywords you are having nowadays. So this picture would give you the impression about the issues of this extensive research. You look at it, you try to understand the situation we are making. I don't know. In West Bengal, we have yesterday possibly 14,000 around positive cases. Throughout the world, possibly we have more than that amount of research article published yesterday. So this is the kind of situation that the pictorial situation is actually growing rapidly. So the amount of research. So what is the issue? The evaluating the quality of COVID-19 research is challenging, even for the scientists who are working on it. You leave the layman, you leave the common people. But even for expert scientists, this is very much difficult. And many are released as preprint, as I was telling, without peer review. And of course, there is another thing is called news media, misinterpreting things. Others are hyped with big press release that overstate the result. But when scientists are finally able to dive into the research, sometimes the study is not groundbreaking as it seemed. This is another dangerous thing that we have encountered. I am honest enough to declare or saying that even for our genomics research, it came into print media, electronic media. Some of the things were very much correct, informative, but exaggeration and the mis-illustration is something which I did not like. Bengali people would understand there is something called dustu gene, clever gene, this ORF3A. This was mentioned in a print media and news media. I did not personally like that particular term because nowhere in the article we said something so. We said this is a special gene having special behavior. That's why you took it. There is nothing called dustumi or dustu things. We did so. Anyway, so that's another issue. So in a science article, science news, actually, it was there that one scientist said, he was a virologist, studying that he could keep pace, he was hoping that he could keep pace with growing torrent of new scientific papers about the disease and the novel coronavirus that cause it. But there are just too many, more than 4,000 alone last week. I mean, there he commented. So it's not possible. He said he is not keeping up. People don't have time to read through the entire articles and figure out what is the valuable things are added and bottom line and what are the limitations. It is very difficult to get into it and extract these informations. So this is a scientist from JHU, Johns Hopkins University. So there are such kind of comments are written here. And so we must concentrate on the quality or not the quantity. So at JHU, they have a very good initiative. They took it they did a curation and they put a curated 2019 novel coronavirus research compendium, which having 80 papers, on 8 topics, including vaccines, pharmaceutical interventions, and they also have written the summary of those articles. So they are trying hard, but you see, you really don't have control towards it. How many articles would you do this kind of things manually? It's very difficult. So it's a very challenging thing that is open to us. So the DOI of this news is given above. You can go into it and details description of this kind of issues. So my layman non-technical suggestion, based on scientific keywords, classify the published article broadly. That is the primary thing we could do, but that we could do in a very technical manner, efficient ways, which Professor Vidyarthi and Anindo, you people must know better and can enlighten in a better manner. But that is what to me is being observed. And make a coincide synopsis of the findings of the article from respective abstract of the articles. That we have to do. For this, you might need technical expertise also to make the synopsis better. And make a database where for each keyword, you get a synopsis and associated references. So if you give me a database of this kind, being a scientist working on COVID-19 would definitely help me to get into those things. So this is my primary suggestion. I don't know how good or bad it is, but this can be taken forward to discuss. So as I said, at the last, I would put a positive words towards it. So this is the vaccine protocol process whenever you require it is. So you have preclinical testing that is done on mice, monkeys, for human thing, because we know mice genome and human genome are 99.99%, I mean, identical. So mice is not much different genomically, at least, from me. So that is done usually, that you do, and then you get into the phase 1, phase 2, phase 3 trials on different levels, and then you get the approval. And this process is very much complicated, and most of the vaccine things are getting failed in the phase 3 trial. And approval is a big issue. It has different aspects: cost, the amount of coverage, the amount of clinical survivability, so many issues. So I'm not a technical person to talk about all this, but this is the complications. You must understand that it is not as easy as you can get it. You don't expect very easily, because we of course we don't know this amount of details of designing this virus vaccine, but it is involved. We must understand, we must give time to scientists who are working day and night to get a new vaccine. So this is very much complicated procedure, because you understand at the end, we the poor people, common people, would use this vaccine. So the entire nation would go for use it. So this is very much complicated and ethical. There are so many kind of approval is required before it get into a door clinic. So, but we have, as on yesterday's data, the preclinical stage, we have 135 vaccine things, phase 1 14, phase 2 11, phase 3 4, and approval also given for one for the military personnels. For the limited use, it has been approved. So this is the very present scenario of vaccine. And as we get an approval of one vaccine, and if it works well, possibly you would get sometime in 2021, middle of that. So that's the positive hope I could see, and I would transmit these things to all of you. And here it is. There are some books to read. Molecular Biology of SARS coronavirus, Corona Replication and Reverse Genetics, Viral Genome, Mathematics of Genome Analysis. So these are the books you could go into it. Topological Data Analysis of Genome for Genomics and Evolution Research. I mean, these are something called for mathematician, fractal geometry in biological systems, these statistical genomics, computational biology. So there are so many disciplines are involved in this particular kind of researches, and these are the books available for reference. We could go through it and we get into it and we discover something new, and that we have to do. And these are the two recently articles published by our team. So those are also given here. If you wish, you can get into it, and wherever you feel difficulty, or interact with me, please do drop me a line through email as given in the first slide, and we can of course discuss things. So thank you, organizers. So I am just leaving it to the organizers. So I am just stopping this.
Thank you very much, Dr. Sarif Hassan, for his wonderful inputs, and he has also shown the research gaps. And for the information science professionals, he has also showed up the open problems and the challenges also. So we are very much indebted to his inputs. And anyone who is interested to interact with him, we have his email ID and phone number also. If possible, you can collect from us, and we will share the PPT to every email. Okay, so I am now requesting Orpita Bhaduri, Assistant Professor of Commerce, sorry, Computer Science, Assistant Professor of Computer Science, to introduce our second speaker and further continue the session.
Thank you, Anindya. I want to introduce Dr. Vidyarthi Datta, who is a faculty member at the Department of Library and Information Science of Vidyasagar University, West Bengal since 2012. He worked as assistant librarian at Shambhilari Mahavidyalaya, University of Calcutta since 2002, and as librarian at St. Xavier's College, Kolkata since 2004. He is an honorary teacher at Bengal Library Association, Kolkata since 2007. He received master's degree in physics from University of Kolkata, associateship in information science from National Institute of Science, Communication and Information Resources, and master's degree in library and information science from IGNOU. He received doctoral degree PhD from Jadavpur University, Kolkata in 2008. He completed one research paper on preservation and digitization of rare documents of St. Xavier's College, Kolkata, funded by UNESCO during 2006 to 2008, and one UGC-sponsored innovative research project on development of information retrieval tool in 2014. Currently, he is working as principal investigator of a DST-sponsored research project on Scientometric analysis of contribution to physics by Indian scientist. Dr. Datta has authored more than 50 articles in several peer-reviewed journals and presented papers in several national and international seminars, conferences, and workshops. Thank you. Over to Dr. Datta. Hello. Dr. Datta.
Hello.
Yes, please.
Yes.
Yes, you are audible.
Yes.
Yes,
Okay.
thank you.
Thank you very much. And it is my pleasure to be present here in this August gathering. And I am highly thankful to Maharani Kasiswari College and thankful to Aninda for arranging such a beautiful August gathering. And I am highly thankful to my previous speaker, Dr. Sarif Hassan, for your really mind-blowing and thought-provoking lecture. Very, very thanking you to Dr. Sarif Hassan. Okay, so let me now, can I start now? Okay. Is it visible? Is it visible?
Yes, sir, it is visible.
Okay. So very good afternoon. research trend analysis of COVID-19, a multidisciplinary and scientometric approach. Actually, my previous speaker covered the hardcore approach. Actually, he covered the core of COVID-19, what actually the virus is, what is its nature. And I will just cover the peripheral areas of COVID-19 because as my previous speaker covered the kernel and inside core of that virus. COVID-19 pandemic, and as my previous speaker also mentioned, it is just today's data in Scopus, which indexed in all 24,453 research articles. So Scopus indexed 24,453 research articles, of which only 19 articles were published in 2019 and rest more than 24,000 articles published in 2020 in this year. And this figure I think would be more than doubled because 2020 just 6 months have passed and still 6 months left. And at the end of the 2020, this figure may reach more than 70 or 80,000 or even lakh. And Dimension database, it indexed even more number of articles. It indexed in total 69,707 articles on COVID-19 published in 2020. 126 articles published in 2019 and only 12 articles in 2018. Actually, in 2018, there was not COVID. I think it was somehow misindexing. Okay. So Dimension indexed nearly 70,000 articles only in 2020. So this is the volume of COVID-19 articles. And if we look at the research trend analysis in Scopus, we can see that in 2020, they index nearly 20,000 papers, 19,972 articles published in 2020. And in 2019, 813. So these are actually the articles about coronavirus. Previous was about COVID-19, it was about coronavirus. And this is the graphical pattern since 2004 to 2020. The sudden hike in the year 2020 is clear. And the first article, it is very interesting to note that the first article on coronavirus indexed in Scopus was published in 1964, while the first article on novel coronavirus, which was a mutated form of coronavirus, indexed in Scopus, was published in 2003. Hence, from the Scopus viewpoint, it may be asserted that the year of birth of coronavirus is 1964, whereas the same for novel coronavirus is 2003. Let us now have a look to the positive COVID cases of India since March to July. Actually, every day when we look at the www.covid19.org, covid19india.org, we can see the number of active cases, number of daily new cases of India. And in India, the first case was reported in 31st January in Kerala. After that, during February, very little, total 5 cases were reported, and again, since 3rd March, the actual in India, the starting or initiation of COVID positive cases started since 3rd March 2020, and at first we'll present the growth pattern in segmentized form, that is the growth pattern for each month for March, April, May, June and July up to 11 July, and then we'll present a consolidated figure. Now if we analyze the growth pattern of March only, it shows an exponential growth. Y is equal to 10.1 e to the power 0.179 into t. Now if we do a prediction on the March data, we find at t equal to 100, that is t equal to 100 means on 10th July. As it is March data, it ends on 31st March. So on the 100th day, starting from 31st March, that is on 10th July. And this data, here the unit of time is chosen as day. Then we find y will be equal to more than 59 crores, nearly 60 crores. But this model already fails because 10th July, just day before yesterday, today in India, there are nearly 9 lakh active cases, most probably, quarter to 9 lakh it may be. But it was nearly 59 crores. So the growth pattern during March, it was very strongly exponential. Next, let us say the pattern of April. Now, during April, the growth pattern shows the parabolic curve, y equal to ax square plus bx plus c, this type of curve, that is parabolic curve. And based on April 2020 data, again, after 100 days, that is on 10th August, the figure will come around 2,93,000, nearly 3 lakh, but it still fails because we have already crossed 8 lakhs. Again, the model for May. During May, it was still exponential curve with very high value of the initial constant. 35581 into e to the power 0.054t, which predicts still at 100th day, that is on 10th September, the total affected number will reach, in case of India, around 78 lakhs, more than 78 lakhs, nearly 79 lakhs. And only time can say what will be actual, and this is very horrifying figure because we have today just 12th July and we have already crossed 8 lakhs. We are just very fast approaching to 9 lakhs. Hence it is very horrifying figure. Again, when t will be 200, that is still 100 days later, on 20th December, the figure will be 174 crores, more than 174 crores. But it is an absurd figure because it will exceed India's total population, that is 138 crores. Yes, which means here comes the de-solar price concept that exponential growth cannot last, which is also which was also predicted by Malthusian theory of population growth, Malthusian demographic theory, that exponential growth must have a ceiling. Next, June. The growth pattern of the month June, it still shows exponential growth, 18871 into e to the power 0.037t, and the predictive model shows 100 days later, 100 days later. That means on 10th October, the total number of affected persons for India will reach around 7 lakh. That already fails because we already crossed this figure. And again, for July population, actually for July population, the correct statistics is not still available because we got only 11 data. Today it's only 12 July. So 1st July to 11 July. And it shows a linear growth. Y equal to x plus b. And on the basis of that figure, July data, we find that 100 days later, that is on 20th October, in India, the total number of affected persons will be 24 lakhs. Still a very horrifying figure, as we already crossed 8 lakhs and approaching 9 lakhs. So these are the results of segmentized growth of COVID cases in India. Now, if we see the overall growth pattern, starting from 3rd March up to 11 July, that is during the entire 4 months, nearly 4.5 months, March, April, May, June, and almost half July. Then we'll find this logistic graph, that is y is equal to a by 1 plus b into e to the power minus ct. And here lies the silver line actually, and logistic curve, exponential curve is continuously increasing, but logistic growth is actually, it is the exponential growth with a ceiling after a certain point of time. And here the negative exponent e to the power minus ct brings the silver line for us. And in this figure, we have calculated the values of the constants, which are A is 1790000, B 763, and C 494. And on the basis of this overall prediction, at t equal to 100, that is just 100 days later, starting from yesterday, it is on 20th October, y will be equal to 17 lakhs. Still the figure is horrifying, but one thing here is clear, that this case, COVID positive case, is gradually towards the saturation. Actually, this negative exponent pattern, this negative exponent factor in the overall growth pattern, it is the silver line. Hence, neither exponential nor linear, that is the overall pattern, but logistic model, and that is the silver line and green hope that that day will come, we can say, when everything will be normal, and we are eagerly waiting for that day. Now here comes actually, now I want to introduce you with this person, John Derek Disola Price, who was a physicist and a historian of science, and actually this person is known as the father of conceptual scientometrics. And once he estimated, around in 1960s, he estimated that the number of the scholarly periodical titles being published by the end of 20th century would exceed 1 million. Although the actual figure crossed his estimation. However, hence today, as the number of COVID cases and also the number of COVID literature increasing at a very quick rate. And in 1960s, 1950s and 60s of last century, De Sola Price came and he showed the way how to provide an effective bibliographic control to manage the information chaos or documentary chaos. And the reason for such chaos is the information explosion. So today we need another Derek De Sola Prize. So how to control or how to manage information on COVID-19? That is the point. And Price also found out the following growth characteristics, that once in 15 years, the number of scientific journals doubles. He found that once in 10 years, the number of articles or literature in a field, particularly in science, doubles. And 1 in 2 years, the number of 2 websites doubles. And this time span gradually decreased. And since after 2016, this time span reduced to a few months. And during COVID time, if only the COVID-related information is considered, this time span reached only within few days, or in near future, it may reduce to few hours. And this is the graphical pattern depicted by Price. This is the famous S-shaped graph of De Sola Price. This is the logistic graph. Actually, the initial part, this graph has 3 phases: formation phase, growth phase, and saturation phase. And the formation phase and growth phase, up to growth phase, it shows the exponential pattern. And beyond growth phase, when saturation starts, it gradually shows the logistic pattern. Hence, only formation phase and growth phase together shows exponential growth pattern, and the 3 phases together shows the logistic growth pattern. And logistic growth has a ceiling, and that ceiling shows the ray of hope for the global COVID cases. And on the basis of this law, Price developed, basis of this proposition, Price developed several laws. One important law is Price square root law. This law also can be well applied to the author distribution of COVID-related literature. For instance, this law says it relates this law pertains to the relationship between the literature on a subject and the number of authors in the subject area, stating that half of the publications come from the square root of all contributors. That is, if 100 papers are written by 25 authors belonging to a subject field, then root over of 25, that is only 5 authors will contribute. 100 by 2, that is more than 50 or more papers. And in COVID paper, we also be able to see the same pattern. And quantitative studies of the network of citations between scientific papers, including the discovery that both the in and out degrees of a citation network follow Powell law distributions, and Price model, a mathematical theory of the growth of citation network based on what would now be called a preferential attachment process. And if only the COVID information case is concerned, the same pattern is still observed today. Now, this is the Price conjecture. Few accounts for many. that is few publications contributed by many authors, or few authors contributed many publications, and same pattern will be observed, may be observed in case of COVID also, that need to be assessed. And this is one of the, this is the most classic masterpiece written by Price, that is Little Science, Big Science and Beyond. This is now open access, freely available in internet. And in the context of COVID-19, we too need today one more Little Science and Big Science. Because little COVID science, big COVID science and beyond. Actually here, little science indicates team research. Here science indicates, it is a figurative term. By the term little science, Price indicated the team research or small group research or small team research that prevailed before Second World War and also before First World War, obviously. And before Second World War, the scientific research was by and large personalized, and it was hardly very big team carried out any research. But in case of big science, the research in very big or in very large team gradually tried to dominate. And this large team research gradually started since after Second World War. That is the point. And Professor C.N.R. Rao, the celebrated scientist, he often called this big science, that is equivalent to bomb science. Actually, the first big science, some historians of science, they tell, first big science started during the time of Manhattan Project, we know. That was the project for making first nuclear weapon, atom bomb. And today, COVID science, it is another big science. Hence, we also today need little COVID science, big COVID science and beyond. And in COVID science also, there are small team research, there are individual research, also there are big team research. And different research groups need to be separately identified. Next, the impact of COVID on different areas. Scopus database showed us that out of nearly 24,000 research articles we searched in Scopus database, that out of nearly 24,000 research papers, how they are distributed over different subjects, and there it was found that around 57% articles belong to the subject of medicine. 15% articles belong to the subject biochemistry, genetics and molecular biology, followed by immunology and microbiology, and followed by social sciences. And that is very interesting point. Social science played very important role in case of COVID case. That is because the COVID, it is a public disease, and public hygiene, these kinds of subjects need direct social intervention, and it left deep impact on the society, on the political scenario, on the socio-cultural scenario, socio-economic scenario, everywhere. Particularly the phenomena of lockdown, which we are facing since 22nd of March of this year. It left a deep impact at every nook and corner of our society. And not only lockdown, there are other features also. The picture of social isolation. Today, if in any housing or in any house one COVID positive is known, the people tries to isolate that particular area. Containment zone or social isolation, these kinds of things bring new impacts on society. And the time has already come to newly think over these kinds of phenomena occurring, and in due course these kinds of phenomena may create the newer social stigma. So next to social science there is nursing, neuroscience, pharmacology, toxicology and pharmaceutics, psychology also, environmental science, engineering, health profession, followed by business management, accounting. Actually in social science also the subject of economics is there, arts and humanities, agro and bioscience, computer science, multidisciplinary studies, also then dentistry, economics, etc. Mathematics and statistics, chemical engineering, physics and astronomy, chemistry. And also beyond those, energy studies, material science, art science, so many areas which are related with this COVID concept. And COVID left very deep impact on our literature and language. There is already so many COVID-19 glossaries available. These terms we hardly used earlier, but today these are very popular, particularly in the context of COVID cases. Asymptomatic, case fatality rate, close contact, community spread, contact tracing, containment, flattening the curve, fomite, home isolation, so many terms. N95, respirator, ventilator, social distancing, pandemic, quarantine, lockdown, and so on. And these are some tag clouds. Excuse me. So these are some tag clouds related to COVID, and there are so many tag clouds available in numbers of bookmarking sites. And here, for instance, obviously coronavirus, this is the most prominent term, but followed by other terms, for instance, danger, outbreak, mask, global, Chinese. This is also interesting. COVID and Chinese, as if these terms get almost synonymous today. Next, COVID-19. Here, the term lockdown also appears with boldness. Quarantine, lockdown, these are also very new terms. This is the tag cloud from a journalist point of view, from reporter's point of view, where the most prominent term is essential rate. Actually, essential rate, it somehow indicates essential market rate, and also it includes the term like essential services. What should be the essential services? And here, the deep impact on social science is found. What are the essential activities? During lockdown period, we repeatedly heard that essential services will remain open, essential activities will remain open. Then how to define essential activities? Every day, essential activities are newly defined continuously. So there are lots of essential activities, and there will be lot more in near future. Hence, these three are very important point also today. COVID-19 and information filter, my previous speaker already pointed out that. How to filter? My previous speaker mentioned that Dr. Hassan mentioned that if today anybody wants to write a review paper on COVID-19, he has to scan 20,000 articles, and I think that is humanly not possible or very hardly possible. And if I have to list the 20,000 articles at the end of a paper, what a horrible situation will come. And so how to filter my information in COVID related to COVID-19. So COVID-19 information explosion and COVID-19 information filtering, from that explosive situation, that is very difficult task, and we definitely need an initial strategy to be formulated to handle these kinds of activities. And next most important feature is COVID-19 and misinformation or pseudo information. That is very, very important, because every day, particularly in every hour, we are facing misinformation, wrong information, rumors, not in the society, even at every locality. We are always listening, we are always hearing. That is COVID positive, that is COVID positive, that is again. And always rumors are there. How to fight with the misinformation, how to fight with the rumors. Actually, misinformation comes where there is no information or lack information, there misinformation dominates. And that happens in case of COVID. Because in case of COVID, as it is a new phenomenon, there severe lacks accurate information, and therefore we have so many misinformation, and hence how to tackle it. And next, another important thing will come, that is particularly interesting for us, that is for information professionals, COVID-19 and plagiarism. There are more than, as Dimension recorded 70,000 COVID-19 articles. Scopus recorded 24,000, more than that. Say I am accepting that 24,000. Out of 24,000 articles, how many are original? How many articles are originally written? And how many plagiarized? So how to check the plagiarism? And it is another huge task. How to find out the exact grain of sugar from a huge heap of sand? That is the hard task. And that is a very challenging today. And next 1 decade, I don't know, at least up to 2030, we will spend time fighting with these issues. So next we will discuss about Scientometric indicators. Now, there are numbers of Scientometric indicators we know. For instance, at first, for Scientometric indicators, let me first just point out the bibliometric approach. And S.C. Bradford and A.J. Lotka, what we know, they are known as fathers of bibliometric approach. I already mentioned the name of Derek D'Sola Price in the context of growth of literature study. And besides the Derek D. Sola Prize, S.C. Bradford and A.J. Lotka, actually D. Sola Prize, he is known as the father of conceptual scientometrics, and Bradford and Lotka, they are known as father of theoretical scientometrics, and Eugene Garfield, known as the father of applied scientometrics. So this is Samuel Bradford, and Bradford's law, that is a very interesting law in scientometrics. That is actually one of the basic law in scientometrics, and which describes the core source items or core journal in any subject that can be applicable in case of COVID-2. This is just one case, it is represented. Suppose if there are 390 articles in any subject, and if we just divide that number of articles in 3 equal segments, 130 each. For the first group, there will be found 3 journals contain first 130 articles, 9 journals contain second 130 articles, and 27 journals contain third 130 articles. And those 3 journals are known as core journals. And similarly, the next 9 journals, those are allied journals and allied zone, and the remaining 27 journals, those are alien journals. Now this is our fundamental task, to find out the core journals. What are the core journals in case of COVID? Yes, Nature is one of them. Nature, Science, those are actually interdisciplinary journals. But one point to be mentioned, the COVID area is highly interdisciplinary, is Nature. Today the situation is so complicated, we can't say that yes, the subject COVID it is a topic under biology, but its one facet comes under biology. But it is a highly interdisciplinary subject with social science, with statistics, with mathematics, and many more. Next, Alfred J. Lotka as Bradford. Bradford's law helped us to select core journals of a subject. Similarly, Lotka's law helped us to select core authors of a subject. And this is the essence of Lotka's law. And from Lotka's law, we can find out the not only core authors, the core research group exist within the domain of a subject. And Eugene Garfield, he is known as the father of applied scientometrics. Actually, today what we see the Web of Science or Scopus, and father of all these citation databases was that gentleman, that is Eugene Garfield. And he introduced some classical scientometric indicators. That is already known to us. Number 1, impact factor, introduced by Garfield in 1975. Next, normalized impact factor. Actually, impact factor is essentially based on citation data. Impact factor is a kind of average, number of citations divided by number of articles. But another point is the citation accumulation speed or citation accumulation potential is not identical for all subjects. That is one point, because citation flow is not equal for all subjects, and therefore the impact factor varies over subject very highly. And to reduce this biasness, in 1987, the Indian scientometrician B.K. Sen formulated the normalized impact factor. Next, aggregate impact factor. It was the impact factor formulated within a subject domain. Next, immediacy index, introduced by Garfield in 1975. And next, cited and citing half-life. And half-life study is another importance. One thing we have to study in near future, that how long the craze of COVID will remain. This is the time of COVID and we don't know 2020, this is 2021, 22, 23. And after few years, say after 2023 or 24, we have to study the half-life of COVID literature. That how long the craze of COVID existed. That is the point. Next, journal impact factor we know, but there are many drawbacks of a journal impact factor. And so, because it is totally citation based and we have to keep it in mind, and it includes self-citation, that is articles in which the article cites other papers in the same journal, journal self-citation, author self-citation. It includes network citation, it includes recitation. So impact factor, it is solely dependent on citation. That is the one negative thing. There are other journal ranking efforts like eigenfactor.org. This is the definition of EF for eigenfactor score and AI for artificial influence score. AI equal to 0.01 into EF. These are formulated on the basis of citation matrix. This is the eigenfactor.org site from where EF and AI data are obtained. And next, H index. What is very popular, excuse me, H index, it is another path breaker and it is today's most popular scientometric indicator, H index. And it was started in the year 2005 and still, but one thing is, H index is still not a complete scientometric indicator. It has so many drawbacks, but in spite of numbers of shortcomings, H index still is highly dominating the scientometric world, because H index alone is paved the way towards the new research in scientometrics. And H index alone paved the way towards citation mapping or indicator mapping, which is the new area. And therefore, even today, in the era of altmetrics, H index was introduced in 2005 by Hertz, and altmetric or alternative metric, altmetric indicator was introduced by Jason Priem in 2010. But still, even after 10 years of altmetric, still today H index is very active research topic, because H index paved the way towards new research. But still, another minus point is, another drawback is that it is solely citation dependent. And therefore, all these points should be addressed. And there are certain databases like Web of Science or Scopus or Publish or Paris, Google Scholars. All these databases, they automatically calculate H index. And there are different ranges of H index values. H index also, it differs over different subject. It differs over subject to subject. For instance, in case of bioscience, its value is generally high. But in case of social science, the 15 or 15 to 20 H index is very standard or very highest lab value. And this is the graph which essentially describes what H index is. Actually, H index is formulated on the basis of number of articles receiving number of citations. This is first H papers received 8 citations. When H number of papers receive 8 citations, this dimension is paper. Along x-axis we plotted paper, and along y-axis number of citations is plotted, and the dimension of citation is also paper. One citation means one reference, that is paper, and this square, its dimension is paper square, paper into paper, hence H square. Hence total number of citation. When first H paper will receive at least 8 citation, this kind of square will be formed, and that indicates the value of H course citation, and square root of this number of H course citation indicates H index. So citation count, publication count, and time, these are 3 fundamental variables of H index. That is the important point. Citation count, publication count, and time. And R is equal to, these are the basic equations of H index. And this is actually relationship between, these are actually empirical relations. This is the relationship between total number of citations N and H index. And this is the relationship between H index and the time in here, where HT will be approximately BT. And for a successful scientist, HT will be of the form of BT. Now for a successful scientist, this is an empirical formalism. For a successful scientist, the value of B will be 1. And for an outstanding scientist, the value of B will be 2. And for an unique scientist, this value of B will be 3. So for any scientist or for any author's H index, the relationship of the variation of H index values with time, here time indicates the length of author's career, that will be like that. HT will be of the order of BT. Next, Van Eyck and Waldman, they generalized the H and G indices in 2008, that is H alpha index. A scientist has H alpha index equal to H alpha if the top H alpha of his or her in publications from the ranked list have at least alpha into H alpha citations each. If H alpha equal to 1, then H alpha will be equal to H. And the H alpha index will be the following property, that is, it has a limiting approach actually. The limit alpha tends to 0, H alpha it tends to P, and limit alpha tends to infinity H alpha it tends to C, where P is the number of papers published by the scientist that have been cited at least once, and C is the number of citations of the most cited paper published by the scientist. Those numbers can be called P indicator and C indicator. Actually, it includes any scientist, they have a huge number of papers which receive only 1 citations. Therefore, C is the number of citations of the most cited papers. Few papers received very large number of citations, where P is the number of papers published by the scientist cited at least once. And there are very huge number of papers received just 1 citation. So the H alpha index tries to bring a balance between highly cited papers, few highly cited papers, and large low cited papers. That is the point. And this is normalized H index, where H index is divided by number of publications. There are tapered H index, and there are so many kinds of H index. That is, H index may be amended or may be changed according to need. Then comes M index, which is the median number of citations received by the papers in the Hertz code. And it focuses on the impact of publications with the highest citation counts. Next, time dependent H index. That is how H index depends on time or at all. It is also very empirical formalism. And then next M2 index, which is equal to H equal to 10 to the power alpha into A9 to the power beta. And from Hn line, this is a log-log straight line curve. This is actually power law equation, and taking logarithm on both sides. After taking logarithm and doing linear regression analysis, the values are found from where the exact indicator value is calculated. And this is the short description of M2 index. And Leo Eghe, he furthered another, Leo Eghe developed further an idea with Glanzel and Schubert. And the idea of Eghe was to base the characteristic scores and scales on the H index instead of on the average number of citations. Next, Ledet-Ledetzow formulated perfectionism index. PIX index, I am just touching this very short. Then also extreme perfectionism index, EPIX. Hence there are PIX and EPIX, that is the comparative feature, and how they are related, PIX and EPIX. Then academic trace index, it was also proposed by Ledet-Letetzow in 2014. And so H index may be amended in different way, but still H index is an incomplete indicator. It is not complete because H index is highly biased and it includes top few articles. If we look at the Scopus or Web of Science or Dimension database for COVID-19 cases, we can find same thing. Very few journals, very few articles which received very huge citations, and once an article receives high citations, it fall into the citation network. That is the point. And H index always hovers. H index hovers within that citation network, and therefore it is biased. And at this point, the E index is defined to complement the H index for the ignored excess citations. And E index, it is formulated like that, which is where E square is the total excess citation, it is equal to C total citation minus H square. Difference between total citation and H square. And from E index, in an extreme case, when E equal to 0, E equal to 0 means for which is highly unlikely in reality, and it completely describes the citation information for papers in the H code. Otherwise, when E not equal to 0, the H index always loses citation information, which is complemented by the E index. Therefore, H index ignores excess citation, and that ignored citation is complemented by E index, and therefore H index can be said to be a complete indicator only when H index and E index are together mentioned. That is the point. Otherwise, this is just from discrete to continuous expression of E index, replacing summation by integration. This is the diagrammatic presentation. Now, the shaded area, that is E square and that is H square, and H square completely ignores this upper part. And that is the main drawback of H-index. Therefore, H-index should be mentioned together with E-index. That is the point. Otherwise, exact indicator cannot be predictable. And this is the standard rectangular hyperbolic curve of any EH diagram or citation article diagram. And next also we can define R index. R index is nothing but it indicates the square root of total number of citations, and this is HE plane. This plane is very important actually for COVID articles, because COVID is an abrupt case, and therefore the manner of accumulation of citation here is very abrupt. Actually, citation is a function of time. With time, it is gradually accumulated. But as COVID is an abrupt case, it is a sudden thing. And therefore, the way through which the citation is accumulated, that is very abrupt. And that is very asymmetrical. And therefore, in COVID cases, we may find for some papers, there may be very high excess citation, there may be very low excess citations. So we have to find out what actually the picture is. And just next I will go to another picture, the centrifugal nature of citation. Just a minute. Okay, I think that is in latter. The centrifugal nature of citation. So that is the important, that what is the relative HE values for each articles. What are the relative HE values? And together, the H index and E index, when these values are mentioned together, only the actual scenario will be known. And R index and A index, they are also associative index to H index. And when we know, then another index is G index. G index is nothing. It's calculated based on distribution of citation received by a given researcher's publication, such that given a set of articles ranked in decreasing order of number that they received, and it is the unique largest number, such that the top G articles received together at least G square citation. So in case of H index, H papers received H citation, and here top G papers should receive at least G square citation. So together is counts. Hence G index is more accurate than H index. And these are the values, and G index is obviously greater than H index. Here, the comparative values of H index and G index for some top scientometricians are shown. This is the GH ratio. Obviously, this ratio is always greater than 1. And citations, it is of highly centrifugal nature. And the question of recitation, inference citation, network citation, these three things are most dangerous. Recitation, receiving citation from one particular person repeatedly, receiving citation from one particular group repeatedly. Previously, I can malarrange a group of people for receiving citation under table understanding. Everything is known to us, so everything can be managed, and therefore citation can also be managed, influence citation or network citation, as everything is managed if we have a source. Similarly, citation also can be managed, and today there develops a citation industry. And if sometimes that citation industry does not necessarily always look at the quality. If any quality works or quality research cannot satisfy any citation industry group, then it may be retained outside the sphere of that citation industry and may not get citation. And people will say, yes, you are not getting citation, your impact factor is low, your H index is low, your quality is poor. But that is also not the case of always. So one thing we have to keep in mind, judging quality is something more than counting only citation or calculating only H index. Judging quality does not mean calculating only H index. That is the point. Particularly the question of recitation, influence citation, and network citation. And also nowadays, the different aspirants, we have pressure. We have UGC pressure, we have API score pressure, we have promotion pressure, we have to publish articles in a hurried mode, and we need citation. Therefore, recitation, managed citation, influence citation, everything is there. Hence, judging quality, that is different issue. And in COVID cases, and this is an abrupt phenomena, the problems of recitation, influence citation, or network citation will play still dominating role. That is the point. Here is just mention that that is regarding actually citation. The nature of the human mind is such that unless it is stimulated by images of things acting upon it from outside, all remembrance of them passes easily away. Yes.
It is already 1 hour over.
Okay, just I'm finishing. And next is tail citation. That is also another point. Tail citation means the articles receiving only 1 to 5 citations, or low cited articles. That is another point. Low cited articles doesn't always mean poor quality. And so citation loss and citation fall. Citation loss associated with excess citation and citation fall associated with low citation. Hence, H index alone is incomplete. With H index, we need to study the other indices. And this is Richard Feynman. He mentioned there is lots of room at the bottom. Therefore, low cited items may yield another big portrait. And uncited versus no citation study, those are important. And once uncited paper, does it mean zero quality paper? Who will judge? And Einstein once made a beautiful statement: as far as the laws of mathematics refer to reality, they are not certain, and as far as they are certain, they do not refer to reality. So judging quality only by counting of impacts may yield uncertain or unpredictable results that may not match reality. And this is Milton's famous statement. They also serve only who only stand and wait. And beyond scientometric indicators, we have article level indicators or altmetric indicators today. So it's all.
Thank you, sir, for your inputs, and you have shown an extraordinary feature of the COVID-19 research in recent times. So, I am now requesting our principal to summarize the whole session, and the question and answer session will be after that. Over to you, madam.
Hello.
Yes, you are audible, madam.
Okay. Maharani Kasiswari College is very much honored today to have such excellent speakers, and I think everybody has felt it that they are very patient and excellent teachers also. A vivid description of COVID-19. The first speaker actually analyzed its nature and the research ways are left for many people, for many scientists to do, so that the industrialists can get a fast track, very, very fast track by the review by various scientists on the works actually have been done on COVID-19, so that the marketing and reaching common people the vaccine or medicine will be much easier. This Dr. Jaman has explained very nicely, and one word actually brought a nice hope. That word is weakening. He said that the virus is becoming more and more weak as days go. Hence, by the lockdown or social distancing, if we can pass some time, as more and more time goes, then the harm leading towards death will be increasing. And around us, and from the news also, every day, we are getting that information also. Earlier, it was really deadly. We have heard most of them were dying, but not now. And we hope it will more and more weak as in the near future. And we are getting a lot from the second speaker, Dr. Vidyarthi Datto, about the nature of growth also. The first speaker also talked about this, and I think according to the first speaker, I missed to say that it is protein, actually polyprotein. Large number of molecules are there. It is RNA, and hence, as we know, that the poison of snake, that is also large protein, and we need antiserum for that. So some medicine like that and vaccine to prevent that will be very much essential for us. Actually, research on COVID-19 is not only a fundamental research, as we see that it is very much urgent for public service, urgent for everybody in this world. So the research guidance or whatever have been done on that, the present lectures are very much enthusiastic. And I think, as Anindo said, that for next 10 years, the general research, maybe other subjects, will be little bit difficult to continue, because for survival of the human being, we will have to fight with the COVID-19. And also this social distancing and the lockdown, they have lot of impact, means negative impact in various parts of the society, including affecting it economically. As we see that the industry and business have been hampered a lot. Not only that, yesterday only in a news, we could hear that even some farmers are unable to go to the field. So overall, this human society is getting affected, and we should be very much actually keen to the solution as soon as possible. Again, I thank both speakers from the college, from Maharani Kasiswari College, and also I thank the organizers and all others involved, and mainly the audience who are having so much patience. And also, I must say that the lectures were very much interesting, that since I was a co-host, I have felt that many people are joining as the audience. So thank you all.
Thank you, madam, for your excellent summary. Uh, so I will now request Dr. Sudipta Ghosh, Assistant Professor of Commerce, to start the interactive session. So we have few questions from the participants. So he will actually conduct this session.
Okay, thank you, Anindada. Now, my first question to Dr. Sarif Hassan, it is asked by Sanchita Majumdar, one very pertinent question. If anyone already suffered from COVID-19, is it possible further she or he will be suffered in the same disease? As you said, COVID-19 is a virological disease.
Yes, it is absolutely possible, and there are so many examples we have already seen in different parts of our country, even outside the country. This is affecting. It depends on antigen and antibody, and once that is lost, that antibody against that virus, once that is lost, and it can again affect. So that is absolutely possible, and that is what is happening even in China also. They are reporting the same thing, that the same patient is again affected. That's
Okay.
possible.
Okay, thank you, sir. Next question asked by Sunita Kejriwal to you, sir. Do you
Yes.
think general awareness or cleanliness is only able to combat COVID-19? How is the social life after post-COVID-19 pandemic?
Yes, to the second question, of course, our social life would certainly change. I mean, as we are having nowadays, so it will certainly get changed. So that would certainly affect our previous life and the post corona life. And yes, now the cleanliness and all those things are of course important, but that's not the only thing, because also one needs to be healthy. uh, Actually, in fact, in proper term, one should be very much healthy. Healthy food is required, hygiene is required. Those things has to be maintained. Actually, that is very much important. Along that, of course, you need a healthy environment as well. So definitely cleanliness and all those things are pertinent and important to this. Yes.
Okay, sir. Thank you. My next question to Dr. Vidyarthi Datta. It is asked by Olka Mistry. Fake news or pseudo information related to COVID-19 is
Yes.
being generated.
Yes.
How one can know the real fact or actual statistics in the context of COVID-19?
Yes. It is very good question. Actually, there are so many information, particularly misinformation or fake news are spreading very fast through social networking at first. Therefore, at first, and though there are filters, okay, now in WhatsApp they provided their filters, they started filtering. Facebook, yes, they also started filtering, but yet we have to at first refrain ourselves from sharing and forwarding message without thinking or without just verifying the same. Actually, many of us, we have habit, just one news, if it is very eye attractive, we instantly shares through Facebook or instantly shares through WhatsApp. That is one of the measure. And next, after getting a news, at first, we have to, there are authentic sources for news, the sources information, and there are various media monitoring services, and it is better to verify each news item with any authentic news sources, starting from NDTV is there, or BBC is there, which are very authentic news sources. Even I tell not all news media are also authentic. For instance, in state level, in Bengal, there are so many news media. There are so many news channels, but news channels are also biased today. And only way is repeated verification of a news and not to do hurry. After getting a news, we have to wait and we have to verify it and not instantly sharing, particularly in Facebook, because Facebook is a very dangerous platform in one way. And instant sharing, we should refrain ourselves from instant sharing. That is one point, I think.
Okay, sir. Thank you. Sir, another question asked by Shayan Roy. I think it is very, very important question. It is asked to you again. Sir, are there any free databases available for data analysis of COVID-19 like Scopus or Web of Science?
Yes, Dimension database is there. Dimension. Yes, Dimension database is there.
Okay, sir.
Another is Worldometer is there. We are getting different information in Worldometer. Also, Ministry of Health and Family Welfare, that database is there. And Dimension is just like if you are talking about COVID or Scopus or Web of Science, relating to just comparable to Scopus or Web of Science, Dimension database is the best that is there. And huge COVID information is there, but we have to filter information. That is the point. And another is PubMed. But in PubMed, citation information is not available. That is the point. Because PubMed is more, it is a bibliographic database, not citation database. But Dimension is just like Scopus, it is a bibliographic citation database. And now Dimension database is there, and another CORD database is also there.
Okay.
But
Thank you,
Dimension
sir.
database is very fine. But still, their filtering is very poor because Dimension is very new. It was started in 2018 just, from the end of 2017, even less than 3 years. Therefore, I also mentioned, I found they index so far more than 70,000 articles, nearly 70,000 articles on coronavirus and novel coronavirus. And I don't know, I think there are some articles retrieved due to misindexing also. That is possible because such a huge number of articles, and that is also possible. And today another thing is also happening that is also very alarming. Suppose any social science or any economics article, authors or editors are just putting the word of COVID-19 at the end just to make it attractive. At the end of, suppose I have wrote an article on the view of Rabindranath Tagore on famine or pandemic, or that is different, the literary theory of Rabindranath Tagore, and at the end I just added one keyword like COVID-19. I know it will be then frequently retrieved, because if we see the Google trend curve, it is the hotmost topic today, COVID-19. And this kind of thing, I'm sure, once the COVID will be, just Dr. Hassan said, it is gradually been weaker, gradually this trend will be weaker. And this search pattern, it is actually very impulsive. For instance, from 14th June to 20th June, if you search the Google trend, you then found the very high search trend against the name of Sushant Singh Rajput. That was the thing. So these are very impulsive things. And so that is the thing. So misindexing is there. So whatever citation database we use, we have to keep in mind how to filter my database, and we have to tally it with my keyword, and we have to keep in mind that yes, misindexing is there, and that is very dangerous thing.
Okay, sir. Wonderful answer. Thank you. Due to paucity of time, we are not taking more questions. We have few other questions also. I think that it will be better if Dr. Sarif Hassan and Dr. Vidyarthi Doctor, they share their email in this platform,
Yes,
and
certainly.
participants then ask you directly. Okay, sir. Thank you very much. Thank you, participants also. Now over to Anindo, sir.
Thank you, Dr. Sudipto Ghosh, for conducting the interactive session. So I am now requesting Dr. Chandrima Karmakar to give the vote of thanks.
Good evening, everyone. On behalf of Maharani Kasiswari College, I extend a very hearty vote of thanks to Department of Library, MKC, for organizing this seminar on research trend analysis of COVID-19, a multidisciplinary and scientometric approach. Special thanks to the speakers, Dr. Bidyarthi Dotto and Dr. Sarif Hassan, for gracing us with their important work and sharing with us their findings and opinions today. It was an insightful and informative session on the pervasive COVID-19. Both the presentations emphasize the importance of the correct use of the humongous information available, while simultaneously underlining the importance of research to understand social conditions and exceptional situations like pandemics. I take this opportunity to propose vote of thanks to the technical team and those who have directly and indirectly contributed to this seminar. A warm thank you to our principal ma'am for her enthusiastic support. Heartfelt thanks to our participants for making this webinar a success. I wish you all have a good evening and thank you.
Okay, thank you, Dr. Chandrima Karmakar, for giving vote of thanks. I sincerely thank the principal of our college who encouraged to conduct such webinar, and I also thank Dr. Bidyarthi Dotto and Dr. Sarif Hassan for their wonderful inputs. And we are really grateful that you have also given us some research topics on which the participants and even we can work on. And the information service is a real challenging issue right now, and we have understood from your inputs that there are many research gaps, and we are actually plunging under the debris of data and information. So we have to develop different kind of filters to filter out the information, and we have to remove the errors and as well as the garbage data also. So thank you all. So with these words, I am concluding the whole seminar. Okay, thank you very much, the resource persons, and thank you, madam, for your encouragement. And I am also organizing all the technical teams and moderators who have worked uh, relentlessly over last week, and we are indebted to the whole team. Okay, thank you, and I am now concluding the meeting.